guys, welcome back to Learning with Isha. Today onwards we start talking about deviations from Mendel's principles. हमने देखा था monohybrid and dihybrid crosses में we obtained certain ratios. Today onwards we'll talk about scenarios where we observe a deviation from such ratios. The first phenomena is incomplete dominance. फर्स्ट ऑफ ऑल एन इम्पॉर्टेंट क्वेश्चन इज ये मेंडल के किस प्रिंसिपल का डिविएशन है? वेल, इट इज अ डिविएशन ऑफ मेंडल्स लॉ ऑफ डोमिनेंस. व्हाट इज मेंडल्स लॉ ऑफ डोमिनेंस? वेल, इट सेड दैट दो तरीके के अलील्स होते हैं, डोमिनेंट एंड रिसेसिव. डोमिनेंट अलील मास्क्स द इफेक्ट ऑफ अ रिसेसिव अलील, बट इनकम्प्लीट डोमिनेंस में ऐसा नहीं होता है. व्हेन टू प्योर लाइन पेरेंट्स आर क्रॉस्ड, so the parents are pure line. When they are crossed, what happens that an offspring is produced. But offspring का phenotype जो होता है, it is intermediate. हम normally expect करते हैं कि dominant phenotype आएगा. But in incomplete dominance, a new kind of phenotype will appear. We'll look more about it as we progress through the video. Let's begin with who discovered this phenomena. Well, it was discovered by a German botanist, Karl Correns. Karl Correns काम कर रहे थे Mirabilis jalapa नाम के एक प्लांट पे, जिसको हम commonly कहते हैं the 4 o'clock plant. So what did he observe? He observed that plants having red flowers when crossed with plants having white flowers did not produce red colored flowers. Instead they produced an intermediate, दोनों के बीच में, a pink colored flower. So that's how this phenomena was discovered. Now moving on, what is this phenomena also called? It is also called partial dominance. It is called blended inheritance. It is also known as mosaic inheritance and intermediate inheritance. Now we begin looking at some examples where this phenomena is observed. So let's begin with incomplete dominance in plants. We're going to talk about two cases. One is Mirabilis jalapa. I've already told you it's the 4 o'clock plant. Other is Antirrhinum majus, जिसे हम commonly कहते हैं Snapdragon का plant. Here also red flowers are crossed with white flowers and the resultant are pink colored flowers. Another case is in animals. This is in fact a very important commercial application of incomplete dominance. That is in the case of Andalusian fowl. The third case is in humans. In humans, it is responsible for causing the Tay-Sach disease. So now let's begin with the first case, incomplete dominance in plants, in the case of Mirabilis jalapa. The Mirabilis jalapa plant has two colored flowers, red and white, where red is mostly the dominant color. So what happens here is that there is a male plant with red flowers and a female plant bearing white flowers. Normally dominant का हम genotype लिखते हैं upper case से and recessive का genotype denote करते हैं lower case से. In the case of incomplete dominance, we avoid that, because the white flower does not represent a recessive trait actually. In fact, it represents a mutant. So what I'm going to use here is a different denotation. I'm going to use R1 to denote homozygous red flower bearing plant and R2 to denote a homozygous white flower bearing plant. Now we are going to perform a cross. In the F1 generation, we get 4 offsprings and all the 4 offsprings are heterozygous and have R1, R2 genotype. Now the next step is to perform a selfing among the plants of the F1 generation. So what happens here is R1, R2 is one parent crossed with R1, R2. This is the F1 generation being crossed, being selfed. So I'm quickly drawing a Punnett square. So here let's write the gametes. Now what we get is, here we start writing the genotype. So R1, R1, R2, R1, R1, R2. So let's just make it R1, R2 for uniformity, R2, R2. So now we have the genotypes of the F2 offsprings. Now what is our genotypic ratio? We observe that R1 के plants I have 1. Okay. R1, R2 के plants I have 2. Okay. And R2, I have one plant of this type. So I observe that the genotypic ratio is 1:2:1, exactly like what Mendel observed. So where is the scope for deviation? In the phenotypic ratio. See, I would expect these two to have a dominant allele, therefore to express the dominant phenotype, that is the red phenotype. So according to Mendel's law, इसमें 3:1 आना चाहिए था आपका phenotypic ratio. क्या ये actually होता है? नहीं. We get an intermediate phenotype for the heterozygote. So the R1 is going to remain a red plant. The R1, R2 flowers are going to be of pink color, and the recessive ones are going to remain white. So the phenotypic ratio now becomes 1:2:1 instead of being 3:1. So here we observe that the genotypic and phenotypic ratio are the same, and this is a deviation from Mendelian principles. Now that we are aware of the phenotypic and genotypic ratio, it's going to be best if we understand it at a molecular level also. Now see, what happens is my R1 genotype is producing my red colored flower. How must this be happening in the cellular level? This R1 gene must be coding for an enzyme. This enzyme in turn must be causing the synthesis of a red colored pigment. Now this red colored pigment will accumulate in the flower petal and cause the petal of the flower to be red in color. Now the R2 gene, what must this be doing? This must be a mutant. It must be a variant of the same enzyme but mutated at a certain point. इस म्यूटेशन से क्या होगा? ये एंजाइम या तो बनेगी ही नहीं या तो ये नॉन फंक्शनल होगी, either no synthesis or non-functional enzyme. So in this case we will not have any enzyme. In the absence of enzyme, there is not going to be the synthesis of any red pigment. In the absence of red pigment, the flower is going to appear white in color. Now let's look at three scenarios. The first scenario is R1. Here both the genes will encode for the red pigment. Let's say one gene individually includes 5 units of the pigment. We are just assuming that की units है 5 unit हर gene contribute कर रहा है. In this case we will have total 5 plus 5 10 units of red pigment. Now what happens in R2? In R2 scenario, my enzyme is non-functional, so there is going to be 0 plus 0. Eventually no pigment and the flower is going to appear white in color. Now what happens in the third scenario, which is a heterozygous condition? Two genes R1, R2. This was being formed in the F1 generation. We observe that R1 तो functional है, makes 5 units of enzymes. R2 is non-functional, not making any enzyme, so 0. As a result, I will have only 5 units. I observe that it is half of the amount of pigment being produced in the homozygous condition. Since pigment की पूरी saturation नहीं हो पा रही है, only 50% of the pigment is produced, so we get a pink shade instead of the red. I get pink shade due to incomplete saturation of that pigment. This is how incomplete dominance works on a molecular level. Now let's quickly move on and look at another example of the Andalusian fowl. In Andalusian fowl, which is an important commercial application of incomplete dominance, what happens is basically there are three colors of fowls. One is splashed white color of fowl, second is pure black color of fowl, and third intermediate है a blue color of fowl. This white and black colored fowls are not considered useful and they are in fact called as wasters. The blue colored intermediate type is considered to be an exotic species and therefore it is considered to be useful. What is happening? Let's try to understand. Pure white male लेंगे, इसका genotype हो जाएगा let's say W1, W1. Pure black female है, let's use W2, W2 here. Quickly we'll write the F1 generation genotype is W1, W2. Now we'll perform a selfing. So we're trying to self this. Let's draw a Punnett square. Now that we have the genotypes, we observe that three genotypes are observed, like in all the cases. First is W1, W1. Second is W1, W2. Third is W2, W2. The ratio of them will be 1:2:1. That is, we know that the genotypic ratio is 1:2:1. What is interesting is the intermediate, the heterozygotes will be the one that will be blue colored. They are the commercially important ones. So the phenotypic ratio in this case is also 1:2:1. What happened commercially is, like I told you, these blue ones were considered exotic. So the breeders thought, why not kill the black ones and the white ones and just cross the blue ones. That's practically what we're doing over here. We're just crossing the blue ones. What we still observe is that सारे offsprings blue नहीं मिलेंगे. हमें only 50% of the offsprings blue मिलेंगे. एक आपको white और एक white और एक black भी आपको observe होगा. So the breeders observed that even by killing the white and black parents, they couldn't get all blue offsprings. We now know the reason. It is incomplete dominance. Now let's quickly understand the last example and the most important one. This phenomena in the case of humans. Tay-Sach's disease is a biochemical disorder. It could be fatal and in neonates it may lead to their death. What happens here is that there is a deficiency of the enzyme hexosaminidase A. This enzyme helps in the metabolism of lipid in the body. In the absence of this enzyme, there is a fatal lipid metabolism disorder. So basically it is a fatal lipid metabolism disorder. Now to understand this, we need to understand कि क्या तीन genotypes हो सकते हैं. Let's denote the dominant here by capital S, the heterozygote by capital S, and small s is the homozygote for the recessive trait. Now what happens is these represent S and small s represent the gene that will cause the production of this enzyme. So here we see that the dominant allele is going to produce a functional enzyme. The other allele also produces a functional enzyme. Much like what we saw in Mirabilis jalapa, 5 units and 5 units, 10 units. So this is enough for the body to function and to prevent this disease. Now what happens in the heterozygous condition? The capital allele makes the enzyme, but the small allele is not able to make this enzyme. What happens here is that only 50%, that is 5 units of the enzyme will be synthesized. But thankfully, still the phenotype of the disease will not come here, as this 50% enzyme is enough to cause the lipid metabolism in the body. Who will then be the affected individuals? Well, the recessive individuals will be the affected ones, as there will be no enzyme in the body. No enzyme means there will be no lipid metabolism, therefore the individual would be affected. So this was all about incomplete dominance. In the next video, we are going to talk about another variation known as codominance. Thanks for watching the video. Do consider subscribing to the channel.